Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Janitza Montalvo-Ortiz. She is a professor at Yale, and her research interests include the study of the genetic and epigenetic mechanisms associated with psychiatry disorders. We are going to be talking about epigenetics and mental health today and how trauma exposure and trauma type are related to mental health and what we can understand with those outcomes through the lens of epigenetics. She really investigates how these trauma-related events increase the vulnerability to develop mental disorders later in life. Her mission of her lab is to advance the understanding of genetic and epigenetic architecture as a way to dissect the underlying mechanisms of the complex interplay between genes and environment that contributes to an increased risk for psychiatric disorders. We're going to talk about several of her studies, really looking at psychosocial factors associated with some of those biological age algorithms in U.S. military veterans and a lot of underserved, underpopulation-based cohorts. If you all are wanting to learn how mental health can be studied through epigenetics, this is the episode for you. And now for my guest, Dr. Janitza Montalvo-Ortiz. Welcome to the Everything Epigenetics podcast, Dr. Montalvo-Ortiz. I'm excited to have you today.
Yes, I'm here. Thank you so much for the invitation.
Of course, and I gave my listeners a little introduction to you before we started off here, and I want to start this show off a little bit different and really start out with the question, what has epigenetics done for you? What does it mean to you? It's a really new field, and I'd love to get your perspective there.
Yeah, so this is a very interesting question, and it brings very happy memories from when I decided to start getting into this field. And it was back when I was a grad student. Not a lot of people were doing epigenetics in the context of mental health. And I saw that as an opportunity to develop myself as a researcher. And as the field develops, I have to say, I started to meet people in the field and even senior PIs that has that personality of like curiosity, higher curiosity, like being more, risk-taking people, because it's a field that, has so many unanswered questions, but still yet like promising in terms of addressing huge gaps in understanding the underlying biology of mental health, which has a huge environmental component to it, right?
Mm-hmm.
So I see epigenetics as an opportunity to develop as a researcher. And yeah, it's an exciting, as you said, new field. I would say relatively new, but with a lot of promises. And I'm so happy that I chose that as a career. Yeah, moving forward.
Yeah, that's a perfect answer. I think that puts a lot into perspective too for maybe anyone listening who's very new to the field, or again, anyone who's been in the field for quite some time. I think everyone in this field of study has a lot of respect for each other because we're taking a deep dive into the unknown, but discovering new things. I think, I definitely learn something new, multiple new things every single day, and love digging into these items even further. So that brings me more into, what are you studying now? I know the mission of your lab is to advance the understanding of the genetic and epigenetic architecture as a way to dissect the underlying mechanisms of the complex interplay between genes and environment, that, definition of epigenetics, that contributes to an increased risk for psychiatric disorder. So I pulled that right from your website. I think it's a great mission. Tell us more about what you're studying.
Yeah, thank you. So in my lab, we have two big research areas. One more devoted to understanding, well, better understanding of human genetics in the context of mental health and psychiatry traits, and also the study of epigenetics, right? And these two components do talk to each other. They are complementary. But I'm curious about understanding what's the connection, if they can indeed inform each other in a better understanding of the pathophysiology of mental health disorders, and what makes them unique as well. What's the contribution of genetic variants to health risk, and what's the contribution of epigenetic mechanisms in health risks that are both shared and unique.
Yeah, definitely. Well, what about this is just out of my curiosity as well. What about your upbringing and where did you first learn about epigenetics or hear of it? I wasn't able to find much about your previous studies before you really discovered this epigenetic world. So I'd love to even maybe know that, and then secondarily, what piqued your interest in mental health and psychiatric disorders?
Right. So for the first question, during grad school, I was very into learning about the neuropharmacology overall. But from a perspective of drug addiction, since that was my background when I did research University of Puerto Rico during college. But then at grad school at Northwestern University, Chicago, I was able to expand my horizons in the research field. I remember that very first interaction with epigenetics. Again, that was something new that my PI said, well, there's something emerging and interesting in this. And I said, well, okay, I'll take the risk. I'll learn more about it. And I attended a meeting in University of Chicago. Remember, that was more in the biochemist area, nothing at all mental health. And they were bringing to the table, more knowledge about what epigenetic is. Like I remember, for example, that was the explanation of DNA methylation, which is one of the epigenetic mechanisms that one of the best understood in humans. So basically, it's just methyl groups added to the DNA that can influence the way the chromatin, the compartments of the DNA are organized, and subsequently influencing gene transcriptional processes, whether the gene gets turned on or off, basically, right? So this DNA methylation, I remember in this conference, it was a huge deal that um, biochemists were started to synthesize the protein that were characterized to be involved in that what they call DNA demethylation pathway. So it was for this first time understandable that these epigenetic mechanisms was indeed plastic, was dynamic. It could reverse itself with the interactions with these enzymes. And before that, it was thought to be, just permanent, DNA methylation, something gets methylated, it will be like that, all your life. But here, it was the first characterization of that. And I was like, okay, so they are mechanisms that are explaining the plasticity of epigenetics. And there's also a crosstalk of how well, if we understand how these enzymes play a role in these mechanisms, we can then develop drugs that can target this and then, modulate those changes pharmacologically and could be, eventually be used for treatment. So that was my PhD thesis on basically. So it was first understanding the epigenetic mechanisms that happens through aging. So understanding that changes in the brain at the epigenetic level were not the same when we are very old than when we are young or adult, right?
Mm-hmm.
And those changes that happens with aging, I was trying to understand how that diminished the efficacy of drugs, for example, specifically antipsychotic drugs, and how by complementing antipsychotic drugs with epigenetic modulators like what's called HDAC inhibitors that could augment the efficacy of the antipsychotic treatment, even in elderly people, or in this case, aged mice, which is
Mm-hmm.
the model that I use to test this. So that was my main contribution of my PhD thesis, and then as a postdoc, I wanted to take that into humans, like understand, I guess, studying the translational component of epigenetics, and specifically in humans in the clinics, how can that be transferable? So in humans, I felt that in order to better understand epigenetics, I also need to understand genetics, right?
Mhm.
Because as I said before, they talk to each other, and play a combined role and unique role both on increasing vulnerability of mental health outcomes. But before going to that, I wanted to provide more insight or more feedback onto why I got interested in mental health. Um, so, as a many neuroscientists that shows neuroscience as a career, um, we read our personal statements, they all start with, well, I have a grandparent with Alzheimer's disease, and that actually has turned into a cliche. But yeah, unfortunately, I had well, three of my grandpas had either Alzheimer's or dementia,
Mhm.
and that awakened a curiosity of understanding what's happening in the brain, and what's happening also in a way that can make the brain plastic,
Mhm.
and that even though we go through different processes at the neuronal level, they can be reversed by treatment, by eating better, following all the doctor's recommendations. You have to eat well, sleep well, exercise. So, how those things can help decrease the risk of suffering from neurodegenerative and psychiatric disorders like Alzheimer's and dementia. And I found epigenetics as a way to better understand those anecdotes that the doctors prescribe, and providing biological mechanisms and hopefully also novel treatments on the road.
Wow. What a background. I really enjoyed that. Thank you for sharing that with us. No, that's amazing. And, as cliche, like you mentioned, it sounds, I think we all start with a story. And then if we have family members, especially when their health is being affected by some, again, field, whether it be mental health, Alzheimer's, dementia, et cetera, I think that makes us just more intuitively interested
Right.
in that subject. So yes, unfortunately, I've been affected by Alzheimer's too. My grandmother had it, and it's yeah, just very devastating to go through that
Mhm.
process and see someone who you love go through that. So I've always been interested in that too. I know I have an APO3/4 variant, so I've been more, cognizant of this field and what I can do from a lifestyle perspective to mitigate some of those effects that may happen with age or as I get older. Um, the lifestyle factors, no one really wants to hear it. It's very
Mhm.
repetitive, but reducing the stress levels, eating better, exercising, um, having quality and quantity sleep, those are
Mhm.
all going to play in a role. And epigenetics and genetics is a great way to tell us more about that. So it's just fascinated by your background, and then even the PhD thesis that you mentioned. Would you call that more of a pharmacoepigenetic realm as well, looking into how different
Yeah,
drugs are affecting the epigenome?
yes, if we
Yeah.
can say, yeah, yeah, that will be a nice description. Yes.
Yeah, yeah. I've spoken a little bit about that field on a couple of my podcasts. Haven't done a really great dive, but I think it's going to be really interesting if you're pairing, how our genes can be affected by different pharmaceuticals, and then even our epigenetics as well, because it's just going to help more and more with that
Mhm.
preventative care and choosing the medication or therapy that is right for that person. The more personalized we can get. So, no, thank you for sharing that background. So what I'm getting from all of this in our conversation, you're using really cutting edge approaches to dissect this epigenomic landscape of psychiatric disorders in human peripheral and postmortem brain tissue. You're focused on studying substance use disorders, trauma, childhood maltreatment, post-traumatic stress disorder, major depression. So a lot of interesting subjects here. Again, people listening can relate to this, whether through themselves or maybe family members too. So how can we use epigenetics as a tool to learn more about these diseases, these disorders? What would you say the best way we can do that is?
Right. So, as you mentioned before, one of the aims of my lab is to understanding the link between trauma exposure and the development of neuropsychiatry traits. And what's the link there? And we do know that a lot of people are exposed to trauma across the lifespan, but it's in fact a very minority that actually develop mental health. So I wanted to understand the resilience, in that association, risk and both protective factors that can help us um, in designing preventive measures and also potential treatments that can help prevent the development of mental health once we identify a person that is at risk. So it's understandable, mostly in animal models, that trauma do induce epigenetic changes. That's very well characterized in the animal model literature. And my mission is to characterize that further and more in detail in humans. But it's so complicated because trauma exposure is very heterogeneous. It can manifest in many ways and at different times across development. Um, and as I said, some people do are like when exposed to the same level of trauma, some people end up with developing mental health outcomes or disorders, and others don't. So, my mission is to understand epigenetic signatures at both peripheral and brain tissue that associates with those individuals that have a history of trauma, and try to also look at the specificity of these signatures based on the trauma type, trauma
Mhm.
exposure, and in the developmental stage that it happens. So it's a very ambitious mission, um, but essential to better understanding what's the link between trauma and mental health. And we're doing this, as you said, you were doing this in peripheral tissue, looking at the blood, looking at saliva. We're doing this in the brain, postmortem brain tissue as well in humans. And also open to collaborate with researchers working with animal models. So if anyone hears this and it's interested in, they're interested in collaborating with someone doing this kind of work, please reach out. But yeah, because I know there are limitations on when working with humans. There are a lot of environmental factors. There are a lot of things that are not captured in the data. There are a lot of confounders, variations that we're not able to necessarily capture in our single regression analyses, um, and also when we identify something, ways to manipulate that and then identify efficacy of treatment, we do need partnership with people doing the animal model work. Um, so yeah, and also in humans, one of the major limitations is to reach good sample sizes, big sample sizes, because each individual has their own story. And these stories, when putting it together with other people, is then hard to tease out whatever we're seeing in the data, if it's in fact what we're interested in studying, or it's not telling us something else, right? So that's why having big sample sizes in these studies that allow us to correct or adjust or account for confounders is important. So that's why I'm collaborating in consortia efforts like the Psychiatry Genomics Consortium Post-Traumatic Stress Disorder Group, where we collaborate with researchers across the globe that has other cohorts that we can find a ways then to work together, combine cohorts, and ask the same questions, but then in better powered sample sizes. And the same with the brain tissue. So right now, I'm trying to put together a study looking at trauma exposure in the brain, combining 3 different brain data sets and reaching sample sizes of close to 400. So one of
Wow.
the biggest so far. But
Yeah.
yet, I guess, the sample size in this cohort do matter, and we want to have the biggest sample size possible. And also the diversity of the sample sizes is important because, although I haven't talked yet about that, but and that will come later down the road, but it's important to have representation of different kind of populations in these cohorts, study cohorts, to make sure that whatever we find in our studies is representative, regardless of our background.
Yeah, definitely. There's so many different factors to take into consideration when you're measuring epigenetics. So, what I'm hearing from you is we're trying to link trauma, exposure, trauma type, and mental health and understand that connection there. And that's really hard to do, as you were alluding to, because you may have trauma when you're young, but you don't start to really go through those processes until you become older. There's a lot of different exposures that could have happened across your time frame, a lot of different trauma types. So there's not really one great way to measure
Mhm.
trauma, so to speak, right? So I think what you're maybe trying to say, and correct me if I'm wrong, but you're using epigenetics as a way to try and quantify that trauma and see what it means, see what genes are being affected in those
Exactly.
mechanisms of actions.
Yes,
Yeah.
exactly. Yeah.
And I think that's a huge need in this space,
Mhm.
right? Mental health can form, a very large taboo. People are really nervous to talk about that sometimes, but I think the more we can talk about it and discuss it and have conversations like these where we're moving the field forward, it makes for the best types of conversations, right?
Yeah. Yeah.
Yeah.
Definitely.
Something else that I really like from your lab is you also investigate multi-omic data sets.
Mhm.
So, your genomics, your methylomics, your transcriptomics, proteomics, et cetera. But to get a better understanding of the underlying mechanisms and identify predictors or biomarkers of these disorders, you're looking at all of these in tandem. Um, can you explain your approach there a little bit, and then maybe I'll back up a little bit,
Mm.
and maybe even defining to our audience what multi-omics is, the different omics and levels there.
Right. So, when we're studying epigenetics, that doesn't work in a vacuum, right? So, epigenetic mechanisms also interplay with genetic variants, also influence gene transcription down the road. We need to understand, what's the impact of changing these epigenetic mechanisms on gene regulation. Do we have higher expression or lower expression of genes? What happens at the isoform level? What about alternative splicing events? And then, of course, if we follow the central dogma of genetics, over the DNA, we have to understand at the gene level, what happens then at the protein level, how that connects. So changes that we're seeing in gene transcription, would that also have an impact on the protein levels? Whatever is translated, will that make a receptor be upregulated in the brain or downregulated? And why is this important? Like, for example, medications work on receptors, right? So they interact with the receptors and go through a whole downstream effects. And we need to understand if changes that we're seeing at the very end of the RNA or regulations at the epigenetic structure or post-translational modifications, how that all goes down into changes in protein levels. So that's why looking at multi-omics is essential to get the big picture. So not only looking at a single domain, but also how that domain interacts and interfaces with other ones, and how they also, complement each other in following the same mechanisms. And I wouldn't limit that to necessarily only epigenetics, like the epigenome, the transcriptome, the proteome. We also need to understand the phenome,
Mhm.
which is essentially everything environmental that an individual is exposed across their lifetime. The connectome, which is all the brain connectivity that happens in an individual, understanding brain activity and how brain regions connect to each other and talk to each other. Um, and metabolome, so all these other things that also are part of the individual biology, and that could also have an impact on health risk.
Yeah. Wait, did you say the connectome?
Yes.
I've never heard of that omic before, so I'm gonna add that to my list and do a little research on those. So, yeah, when you're incorporating these multi-omic factions, if you will, it's more of that big picture. How do they interplay? How do they affect one another? Where do you see that effect? And I think it becomes really interesting then when you talk about a single omic. How do you measure that omic? And then how do you go from there? So these are subjects in their own realm in its entirety. So being able to connect with people kind of like you were talking about earlier and putting all of this data together can help us make more conclusions on a larger scale. One thing you brought up was talking about a lot of the research you're doing is performed in underserved populations, and that it's important to make sure we have representation and diversity. Specifically, you look at a lot of Latin American populations and military veterans, which will be one of our main focuses today as we talk through some of your publications. And it may be rather obvious, but I love just asking you as the expert here with your background and your work, why is it important to look at these populations, and how can we extend these findings to the general population, which, again, as you hinted to, is our overarching goal?
Right. So my lab, one of the main goals of my lab, it's making sure that our findings are transferable across populations. And I noticed a huge gap in better understanding of it, the human genetics on populations like admixed populations like the Latin American. I'm a little bit biased because I'm Puerto Rican, um, but yeah, and also the complexity of analyzing data from people that has admixed background. So admixed being a mix of different ancestry groups.
Mhm.
Um, and what we noticed is that human genetics field was rapidly advancing. We started identifying genetic variants associated with multiple disorders. Started off with schizophrenia in better power samples, but um, there were still underrepresented populations that were being left behind. And I wanted to address that gap, and also a better understanding what admixture plays a role in also health risk, um, and why that this is important also in epigenetics is because many of these genetic studies, when they do what it's called genome-wide association studies, they look at the whole genome, or genome-wide level associated genetic variants, and they identify sometimes genetic variants, or we identify genetic variants that map into places in our genome that there are like multiple genes in that area, right? So it's not really well understood. Is this genetic variant mapping to this gene, or is it like all these different genes also playing a role in the risk? Or is it a gene that is like nearby and not even in that region?
Mhm.
So how do we address that question? Use, for example, epigenetic data to get a better understanding of, whatever findings we get from GWAS, and then identify the what it's called functional genetic variants. So we look at ATEC sequencing data. We look at to look at the chromatin landscape, going to the notion that where we have open chromatin, we have like a lot more going on in terms of gene regulation. We look at histone modification landscape. We look at DNA methylation, and all these layers of gene regulation. We use that information to better inform the functionality of the genetic variants that we see. So I know in the news people say, "Oh, they found a gene for this disease." It's not actually the gene; it's the genetic variants. And these genetic variants could be higher prevalent in one population, but absent or non existent in other populations. So that's why it's important to do these studies in different ancestral populations to make sure that the genetic variants that we're identifying, they're also prevalent in their population, and that whatever um, benefits could arise from applying this to clinical settings could also benefit populations that are not as well represented. And also, incorporating diversity in epigenetic work or related functional work is essential because the epigenetic patterns could also vary across populations. So we want to make sure that when assessing functionality of genetic variants, we're using the right tools and the most informative tools also for underrepresented populations.
Yeah, I think getting that nomenclature right is huge. I'm sure that drives you bonkers as, a scientist, hey, the genes and then the genetic variants and understanding what that means. But you're exactly right. The more diverse of a background we can get from a genetics and epigenetic standpoint, because we're all going to be exposed to different things, even if we're in a different location. So, even people in neighboring states, neighboring cities, I can I've seen data before that by having a raw IDAT file or looking at someone's raw methylation values, you can even tell where they live down to the zip code based on what they've been exposed to. So really having a diverse group and again, different ancestral populations, I think is something hopefully this community and people in this field are gathering around and putting more effort into. I'm going to switch kind of from talking about your research in a general way to getting more into your papers. So I remember seeing your name first on this paper here. You were looking at the psychosocial factors associated with GrimAge in male U.S. military veterans. Yeah, can you describe this paper to us? Tell us what you found. I think our listeners are probably familiar with GrimAge as well, but I don't think we've ever talked about GrimAge too much in depth. So even if you want to, explain the death predictor a little bit further too, that'd be great.
Yeah, sure. I mean, I did listen to previous podcasts and I did my homework. And I've noticed that there has been conversations on the first generation and second generation epigenetic clocks. But yeah, you're right. Like the definition itself of GrimAge, which is the epigenetic clock that we have evaluated more recently, is basically an epigenetic clock that is meant to be a predictor of mortality per se, because it includes, for example, DNA methylation surrogates of plasma proteins that are associated with mortality risk, and also incorporates DNA methylation based estimators of smoking, specifically smoking pack per years.
Mhm.
So it combines, in addition to CPGs, it combines that perspective or CPGs associated specifically with mortality, sort of like to come up with a predictor of mortality risk, but also incorporate that aspect of smoking, which has, you know, it's a has a huge effect in DNA methylation patterns. And it's something that has been widely consistent across the literature, and also something that has been closely linked to aging and to mortality risk as well.
Mhm.
So it's sort of a unique and sort of different from other epigenetic clocks, and the fact that it's a combination of these two components. That's why I really like it. And we have tested other multiple epigenetic clocks, which I'm sure other researchers have done the same. And so far, GrimAge has been constantly the one that, it has a better correlation with chronological age, and has performed better in our analyses. So in the psychosocial study, we have published two recent studies in the same cohort, but looking at different aspects. And this is in collaboration with Rob Petersak. And one paper, the first author, it's Amanda, who has done an amazing work on putting all the data together, and Peter Na. And I obviously was happy to collaborate with them. So what we did is that in 1,135 U.S. male veterans, we look at the associations of GrimAge with different psychosocial and lifestyle factors. So, and what we found is that when we look at health and lifestyle, when what came up as explaining the most variance of accelerated GrimAge was the number of days engaged in physical exercise. And I have this down so I don't forget the details, because I feel like, the magic is in the details on what we found in this paper. So each additional day was associated with 19% decreased odds of having accelerated GrimAge. So the more you exercise, the less accelerated GrimAge. So the less your biological aging, basically, right? So, and then this was also, we saw also associations with higher accelerated GrimAge with lifetime history of SUD, substance use disorders, and alcohol use, poor sleep, also not be married or cohabiting with someone, with a partner, with greater cumulative trauma exposure. And this is also something that we found using the Horvath epigenetic block on a previous published paper. And also, in terms of psychological factors, we found an associations with lower dispositional gratitude and openness. So people that are more gratitude has less biological aging, basically. So, and I think that's why understanding social determinants of health is important to know, what aspects of our environment, what aspects of our social life, of our nutrition, of our exercise are associated with these parameters of biological aging, which are, also associated with health risk. So that's what's one of the papers. We found that was like a close to 20% of our cohort had accelerated GrimAge. And again, this was linked to being less likely to be married, being obese, being current smoker, suffering from alcohol use disorder or PTSD, and PTSD showing greater severity of symptoms of PTSD and reporting more cumulative traumas.
Mhm.
So, yes.
Yeah. Very
Yeah.
interesting. No, and I think I appreciate you going into the details. I think we need those details, right? And I'll link that paper in the show notes. So if anyone wants to give it a read, I definitely recommend it. But yes, understanding, I think, how those social determinants of health actually affect our mortality, our morbidity, our GrimAge, these biological age predictors. So I think you hinted at it already through noting that. But what else do these results suggest? What are the applications here? Is it really people saying, okay, here's my lifestyle, here's where I can improve? I really thought the gratitude part was interesting too, and the openness. I keep seeing articles like that in my Google alerts that the more you express gratitude, the better your aging is. So I thought that was a good snippet as well.
Yeah. So, like, for example, one of our, like, Rob Peters, who is the collaborator, he looks at this from the clinical perspective, right? Because he wants to look at interventions that we could do in veterans that can help their quality of life and subsequently their health outcomes. And he said, well, this gratitude finding is so interesting because we could just use gratitude informed interventions and hopefully see how that impacts the health outcomes in our veteran population. So there's certainly, therapeutical interventions that could be informed based on our findings, and it will be very interesting to see how that, certain if that indeed can have an effect.
Yeah.
Yeah.
I agree. Are you looking to do an interventional trial in the future, or maybe your colleagues? Because that's what everyone wants to know. I think the better question I should have asked there, instead of what do these results suggest, what's the clinical use, or how can we use this, in our daily life?
Yeah, so before going into a full clinical trial, what we are intending to do is to try to replicate this in
Mm-hmm.
independent cohorts, right? To
Mm-hmm.
make sure that what we're seeing is not, some spurious finding, but in fact, it's a real finding.
Right.
So
And
yeah, next step is to replicate the finding, and then I would say definitely we have clinicians at Yale that are at the cutting edge of designing treatments for trauma populations. So I'm sure will be easy to find collaborators that will be interested in assessing this in this population.
definitely. And I think the replication part of the study, that's just as an important part as any of this entire process. So I'll be excited to follow along with your work and hopefully get to that interventional level too. Um, all right, the next study here. So you've also had a paper looking at the epigenomic differences between smoking and nicotine dependence in a veteran cohort. So you did mention this earlier too. The epigenomic literature on smoking is very well established,
Mhm.
but studies evaluating the role of epigenetics and nicotine dependence are limited. I actually, prior to looking at your work, didn't know anything about nicotine dependence with the epigenetics. So yeah, let's talk a little bit about this study. Can you describe that there and walk us through?
Yeah, of course. Yeah, so one of the main questions that I had, and it's because the cohort that we study, another cohort is called Yale Penn, led by Joel Galenter and Cransley. Um, it's a cohort that we have a lot of people with substance use disorders because it's meant to study the genetics of substance use disorders, and the same cohort I used for the first EWAS that I published in the literature, that is published in the literature, of opioid use dependence, and it was in a woman population. And I wanted to make better use of that cohort. And I saw the opportunity that maybe we can use this cohort to disentangle the question of, smoking signatures from nicotine dependence in a way
Mhm.
to not only differentiate smoking from nicotine dependence, but also potentially finding biomarkers of nicotine dependence, which are greatly needed in the field. But we have the major limitations that smoking is so, the effect of smoking is so substantial in methylation that studying this will be very hard if it's not in a cohort that we have high prevalence of substance use disorders. So I saw this as an opportunity to study this, and this is the cohort that we're expanding the initial findings from the veteran cohort. It's called the NHRBS, and we're looking now at sex differences. But yeah, telling this long story short, um, we used the veteran cohort to look at the differences between smoking and nicotine dependence, which we're now expanding in the open cohort. And what we found is that the genes that were associated with nicotine dependence and not and we didn't found in the smoking EWAS were related to neuronal differentiation. So we have, for example, the NeuroG1 gene is a finding that we didn't see in the EWAS of smoking, but we only see it when evaluating nicotine dependence. We also see two other genes. One of them we see a high correlation with between blood tissue and brain tissue, so potentially relevant in the brain as well. And then by doing co-methylation analysis, which is, different from EWAS analysis, epigenome-wide analysis, where you just do a linear regression and look at the association between each CPG site with the phenotype of interest. Co-methylation analysis looks at correlation between CPG sites that are nearby and how the group of correlated CPG sites are associated with, that is called modules, are associated with the phenotype of interest. And we identified that one of the main modules associated with nicotine dependence was enriched for nicotine addiction, which was, very reassuring that what we're seeing in the analysis with nicotine dependence specifically were informing, in fact, that trait and not smoking. And what we see in that module are genes in the GABA family that are very well known and in the SUD, in the substance use disorders field, as associated with addiction, including nicotine addiction, and also shown in animal models to be involved in nicotine addiction. So I think we did a really good job at disentangling the smoking from nicotine dependence in our methylation data and identifying potential specific signatures for each trait, but essentially more for nicotine dependence, which is what we have in the cap in the literature. And now what we're intending in the Yale Penn cohort is to expand that to a larger cohort and also incorporate women to see if there are sex differences as well.
Gotcha. Very interesting. Super cool. I thank you for explaining that. I have a lot of follow-up questions. Can you just describe the difference between smoking and nicotine dependence as it relates to this study? I think that's important.
Yes. So smoking are just how we treated smoking is current smokers. So people that just use smokers. However, not everyone that smokes suffers from nicotine dependence. Not everyone develops addiction. They could be current smokers now and then stop the week later and then never smoke again. Whereas there are other people that they will be smoking now and then they will just keep smoking and smoking more and then developing a tolerance. So they need more packs per day. And then they will develop withdrawal symptoms. So when they stop smoking, they will start having physical reactions in their body and having that urge to smoke again, having that craving. So when they started developing these symptoms, then they have developed nicotine dependence. So that's why it's essential and it's very important to differentiate between the two because we want to understand, what's related to just exposure to smoke, just exposure to the nicotine, whereas what's more related to the addiction surrounding the chronic use of nicotine.
Yeah, I think that's important. And nicotine dependence as well. I know they have like those patches or, the pouches. Would that fall under the nicotine dependence category too?
Right. So people that are using patches, most likely it's because they have developed nicotine dependence and are using the patches as a way to eventually stop smoking.
Yeah, that makes sense. No, thanks for differentiating that. And then, so you're really looking at the neurodevelopment impacts of addiction by, you said the GABA families being affected. And then you also, did you say you were doing the EWAS, but then you also did the co-methylation? Is that what you said it was called? So you're looking at a group of methylation markers, little clusters that are next to each other to see if they're being affected in maybe the same way or different.
Yes.
Okay, perfect. Yeah. New definition there as well. I'll outline that for people just so they can understand the difference. But I think it's important to differentiate because even with those smoking signatures, I know DNA methylation, you're not going to find a stronger signal between anything else and smoking with that. A lot of the DNA methylation signatures are going to be more accurate than your self-reported smoking habits. So it's you can count the secondhand smoke into that, and it's hard to differentiate. So I think we need to have that nicotine dependence arm of this study as well, and appreciate the work that you're doing there.
Yeah, thank you. I can give some advancements on some research that we've been doing.
Yeah.
So linking epigenetic aging to SUDs, I have a very talented postdoc that is running analyses looking at the Yale Penn cohort that I mentioned, looking at epigenetic aging, including GrimAge. And we're looking at across the board, comparing different substance use disorders. Of course, nicotine dependence was the one that was more prominent as the impact of accelerated epigenetic aging. And something very interesting is that when we look at using self-reported data, when we look at current smokers, never smokers, and former smokers, epigenetic aging for never smokers is very similar to former smokers. But when we see current smokers, the accelerated GrimAge is like huge. So it's
Mm.
very interesting to see, and it is consistent with the literature that once you stop smoking, you have chances to go to
Yeah.
where you were at the very beginning.
Yeah, again, reversible.
Yeah.
So former and, never smokers look basically the same, but current we can see a very large
Yeah.
difference. So it can help, maybe people try and decrease that habit, that use of smoking.
Yes.
Yeah. Okay, I know we're getting short on time here. I have one other study I want to talk about. The last paper is going to be the EWAS, where you're actually looking at post-traumatic stress disorder in U.S. military veterans. So here, I know you identified 6 genome-wide significant CPG sites associated with past month PTSD. So maybe more current, and
Mhm.
I'm sure you'll get into that. And then 3 CPG methylation sites with lifetime PTSD. So yeah, curious to hear a little bit about what you found, what these genes do, and the CPG sites they're related to and map with. What are they involved with? Those mechanisms of
Right,
action.
sure. Yes. So we evaluated PTSD 2 different ways, looking at past month. So people that have reported symptoms of PTSD within the past month, and their lifetime. People that have just history of PTSD diagnosis across, their lives. So in a way to understand, since we're looking at epigenetics, can we identify signatures that are, associated with currently like having or expressing symptoms of PTSD? Whereas, can we still have those epigenetic signatures from PTSD that happened,
Mhm.
a long time ago or longer than current symptoms? So we did this, and we identified, as you said, 6 different CPG sites with the current or past month PTSD EWAS, and 3 with the lifetime PTSD EWAS. And I wouldn't go too much into this because, again, I do value replication in a way
Mhm.
to understand if whatever we're seeing is, actually real. So I would talk about what we replicated. So in the paper, we were able to replicate one of these findings in the graded trauma project cohort,
Mhm.
and with the same direction effect. This is what's with the CNP7 finding, and from those 9, we were more confident of this finding because we were able to replicate it, and it was like in a very mixed also cohort. I think it was African Americans, and the main finding was in European America. So the fact that we replicated and we replicated in a different ancestry cohort, it provides us confidence and also reassuring that maybe the findings are indeed transferable across ancestries, which is an important thing to consider. And then we said, okay, so we're seeing this replication on the lifetime PTSD. We're seeing this in the blood. We wonder, what about in the brain? Are we seeing changes in the gene expression of this gene associated with PTSD in the brain? So we look at data from the VA Brain Bank, and it's the National Center of PTSD Brain Bank. And in a cohort where we have PTSD cases and healthy controls, we evaluated tissue from the orbitofrontal cortex, one of the cortical regions also associated with PTSD in the literature. We found that we see differential expression of this gene in the brain associated with PTSD. So
Mhm.
not only we were able to replicate this finding in an independent cohort and in a different ancestry, but we are also validating this finding in changes in the brain and at the gene expression level. So I think it was a very, very neat story. A follow up on that will be to do it in a larger and more diverse cohort and see if it replicates and potentially identifying novel hits associated with PTSD. And not only in saliva, the samples, but also we're looking at in evaluating human postmortem brain as well.
Yeah, that's great that you had access to that brain tissue and you're able to, look at that even
Yeah.
further. I may have missed it. You said it was the CMP7, is that correct?
CNP7.
And what is that
Sorry,
exactly known for?
SENP7. Sorry.
S
Yeah.
S E
So
N
it's a
P
gene
seven.
involved in transcriptional regulation, but what's very intriguing is that it has come up. Genetic variants of this gene has come up in GWAS associated with risk-taking behavior and alcohol
Oh, yes.
consumption. Yes.
Okay. I remember reading about that now. The light bulb just came out of my head. So yes. Oh, very, very cool stuff you have going on here, Dr. Montalvo-Ortiz. I'm super excited about all of the work that's coming out in the replication studies and just digging more about the connection between mental health and trauma and what epigenetics can tell us about that. So we're almost done with this podcast. I do have a curveball question I always ask everyone at the end that wasn't on the agenda. But if you could be any animal in the world, what would you be and why?
I did tell you I did my homework, so I was expecting
Oh,
it
darn.
already.
Yeah, you already knew.
Yeah, so it gave me plenty of time to think about. But actually, when I heard it, the very first time, the first podcast, sorry, I instantly thought about a bird.
Oh,
Actually, any kind of bird, not like specifically. I'm not very nerdy on birds, but I think the fact
yeah.
that they can just fly whenever they want, like get to a different town, country, and see a lot
Yeah.
of different things. I love travel, so I guess
Hmm.
I see myself as a bird.
There you go. Well, yeah, I forgot that you've listened to some. So maybe I should start asking people like a random question and, I'll make
I do
something
like this
up.
question. I think
Yeah.
you could tell a lot about the personality of the person.
Yeah, yeah, but you had an answer. You thought about it. I love it. A bird is great. Well, I appreciate your time more than anything. I know you're super busy with everything you're doing in this research and everything you're giving to this field. For any listeners who want to connect with you, where can they find you?
Right. So I can certainly share my email on the website that you can disseminate if that will be good. And yeah, I'll be more than happy. People that are interested in collaborating with us, please reach out. And I wanted to thank you, Hannah. I think this podcast is an amazing idea. I think it's a very good platform to bring researchers and experts in epigenetics together. And I had a really fun time to hear your previous podcast. So congratulations on this work as well.
Yeah, well, thank you for those kind words. That means a lot. So, this is definitely a new area for me in terms of like the production, the marketing. I'm not very creative, so it's really fun to have this outlet and really talk to experts like you. So, no, I really appreciate that. And for everyone listening, again, thank you for joining the Everything Epigenetics podcast. Remember, you have control over your epigenetics. So tune in next time to learn more. Thank you so much.
Thank you. Bye bye.